introduce Professor Dai Herbert, who spent a long time in South Africa, but originally comes from Wales here in the UK, to which he has now moved back. And so we see him from time to time at the National Museum of Wales in Cardiff, where he is extremely welcome as not only an expert on South African non-marine molluscs, which he'll be talking about today, but also marine molluscs, including those of South Africa and other places. And he's one of the editors of the mighty MolluscaBase as well, which since it first arrived about 10 years ago has really transformed online taxonomy of molluscs. And he's done many other things too. And he's a really good speaker, and all his work is always top quality. So I'm sure this talk will be exactly the same. I hope you all find it really interesting. So thank you very much, Dai. Take it away.
Thank you, Ben. As Ben said, I'm originally from Wales. I was born and brought up in Carmarthen in West Wales. And then I went to university in London for undergraduate and postgraduate studies. And then I headed for South Africa in 1984 to take up a 3-year position, which ended up at 33 years. I joined Dick Kilburn in the Natal Museum, who was my mentor there for many, many years. And his daughter is here today. And I've worked together also with Linda Davis, many of whose drawings you will see on the slides today. And her daughter is also here with us today with her son-in-law as well. So I'm going to talk to you today about the South African land snail fauna and how this has been affected by the phenomenon of continental drift, plate tectonics, and so on, and to see what reflections we find in that fauna today of that geological history. But before we go on, I want to just put South Africa in its context today. We have here South Africa, obviously at the tip of the African continent, a temperate region, isolated from similar southern hemisphere temperate regions by huge expanses of ocean. And then to the north, it's bounded by the tropical regions either side of the equator. So we've got very much an isolated landmass with a largely temperate climate, isolated from similar land masses in the southern hemisphere. That wasn't always the case. If we go back 150 million years, in the time of Gondwana, when the land continents were in a very different formation, we have here Africa joined closely to South America and to Antarctica, to Australasia, and to India and Madagascar. By this stage, 150 million years ago, India and Madagascar were already starting to drift apart. And then by 170 million years ago, West Gondwana was beginning to, or had already started to split. The early Atlantic Ocean was forming here, and basically Africa was on its own, separated from the other fragments of Gondwana, South America, Antarctica, and Australasia over here. So, but now things look very different. And we find ourselves with South Africa, as I said, very isolated. But the climate there is dominated by oceanic currents. We have the warm water Agulhas or Mozambique current coming down in this region. It's called the Agulhas current, heading towards the Agulhas bank down here. This brings warm tropical water, which makes the climate in this region substantially warmer than it would otherwise be. And then on the opposite side of the country, on the west coast, we have the cold water Benguela current coming up from Antarctica. It's a deep water current, but it upwells off the coast of western South Africa and Namibia, bringing nutrient-rich water to the surface. And so it's a very productive marine environment here. It influences the climate on the land, particularly. There's very little rainfall in this region. And so we have a wet summer rainfall region in the east, and we have a Mediterranean climate in the south here, and then a very much drier climate on the west coast. On the east coast, we have lots of forest ranging from Indian Ocean coastal belt forests to Afro-montane forest in the higher elevations. And then in the Eastern Cape, we have deeply incised valleys filled with thicket-type vegetation. In the Cape, we have the famous fynbos vegetation associated with the Cape Fold Mountains, a very low nutrient type of environment. It's not so good for snails. Certainly, they're not as numerous there, but then on the other hand, the ones you get there are very interesting. In the middle of the country, we have the Great Karoo, which is verging on semi-desert, certainly very, very dry, but spectacularly beautiful. And then on the east coast, on the west coast, we have Namakwa land going up into Namibia, where we get lots of semi-desert and desert type habitats. So I'll start by going to some introductory remarks about the land snail fauna and mentioning some of the Gondwana relics. And we're going to take a look at certain of these southern groups in more detail, in particular the families Rhytididae and Chlamydophoridae. And then I'll spend some time talking about an unusual little genus of snails called Prestonella. So looking at the mollusk fauna of South Africa as a whole, we really only have representatives of the Polypocophora, scaphopods, gastropods, bivalves, and cephalopods. There are no recorded species in these classes over here. The kind of level of diversity that we've got here is equivalent, at phylum level, is equivalent to this level of diversity in the chordates, ranging from fish through amphibians, reptiles, and birds and mammals, to the more smaller groups of non-vertebrate chordates, the sea squirts and things like that. So it's a huge range of diversity encompassed within the mollusca. In terms of the relative diversity in South Africa, don't worry about all these figures here. The totals for chordates are about 4,400, and for mollusks about 4,200. So similar levels of diversity across the phyla. But if you think about the number of people studying chordates as opposed to the number studying mollusks, then it's very, very different. Getting on to the land snails, for the southern African region as a whole, about 630 species, 80% of which are endemic, 86% endemic to the region as a whole, of which 66% are endemic to South Africa itself. Most of them, 72% or so, are herbivores or essentiulato, and detritivores, omnivores, but a surprising number, 28%, are strict carnivores. They really do not eat vegetable matter to any great extent, and the vast proportion of their diet consists of animal matter. So I think that's a relatively high proportion, certainly compared to the land snail fauna of the UK. Some of the more standout families, we have acatinids. I'm sure you're all familiar with acatinids, the giant African land snails. And then the all snails here, Subulinidae, now generally thought of as a subfamily within the Acatinidae. We have the Cheropids, many, many different species of those. Currently, until recently, only thought to belong to 2 genera, but that really is incorrect. I'm sure there are many more genera there, lots awaiting description. Then we have the Cerastids, the so-called bark snails, and the porcelain tree snails. Very attractive little snails, often seen wandering around on the leaves at night. They hide away during the day, but at night time you can go out and see them crawling around. Then the streptaxids, genera like Gulella, huge levels of diversity there. These are carnivores, eating soft-bodied invertebrates. And then Another commonly encountered group are the operculates of the family Pomatiidae, the so-called shuffler snails, because they walk with the different sides of the foot going along like this. They seem to shuffle along. Very quaint snails. Typical thing here is the eye is at the base of the tentacle instead of the tip, typical of operculates. And then another large family are the urocichlids, ranging from true snails through semi-slugs that can't withdraw entirely into the shell to the true slugs, which only have a little vestigial shell here that lies within, underneath the mantle. And then, unfortunately, you can't read the title there. These are some of the southern groups, the so-called relic groups, if you like. And the ones we're going to be talking about today are the Rhytididae, the cannibal snails, and the Chlamydophoridae, the hunter slugs. Other relicts are the Dorcasids. These are confined to the drier desert type habitats and coastal habitats on the western region of South Africa. And the Sculpteriids, another very, very dry climate group, mostly found in Namibia. Namibia is not a very hospitable place for snails. It's so dry there. But nonetheless, there are hardy souls that can survive in such an arid climate. Then there are two other southern groups, which are thought to have been derived from northern groups. These are the Cape slugs, as they're called, Uapeltidae. They were long considered to belong to the Arionidae, which is a European family, but now they're regarded as belonging to a family of their own. And then the Fauxullidae is another endemic family in South Africa, but originally thought to be part of the Orculidae, which are well known in Europe as well. So these two are not really part of the southern relict fauna, as it were, although they are southern endemics. I think there's supposed to be a title here, but so this is the distribution. Can we get rid of that thing?
I don't know why it won't go away.
Is it that layout?
Which one? This
Yeah.
one. No. There we go.
Oh, there we go. Thank you. So the global distribution of the Rhytididae, the first family that we're looking at, this was what was thought to be their distribution until we started looking at it more closely. Not me, but we malacologists. And then it was discovered that these East African ones that were thought to be Rhytidids are actually. Oh, now what have we got? We've got a problem here.
Just click on the screen again. Okay.
There we are. I'm going to see if that works. So the East African ones were discovered to be not Rhytidids, but in fact flat streptaxids. So we can eliminate them. And the same goes for the ones from the Seychelles and so on. They too are now thought to be streptaxids rather than Rhytidids. And this still remains a question mark about Madagascar. We'll look into that a little bit later on as well. So basically, we're looking at South Africa, Southern Africa, New Zealand, Eastern Australia, and there's some rare ones down in Western Australia, and then Melanesia, New Guinea, and so on as well. This is just a glimpse of the diversity across those regions. In South Africa, we have the larger cannibal snails, the Natalina genus, and the dwarf cannibal snails, genus Nata. That was it, just two genera in South Africa. And then in Australia, there's a great deal more in terms of generic diversity. I've just shown two here, Ostrorhytida and Victophanta, the so-called black snails of the Nothofagus forests in southern Australia. New Zealand, we have Paraphanta, Pauliphanta, the cowrie shells. These are quite well known to the general public, and the genus Rhytida, after which the whole family is named. In New Caledonia, there's a bunch of them there too, rather more unusual ones. This one is biconcave, very interesting little snails, and these two rather bizarre looking right-tittids. There are more conventionally looking ones there too as well. So in terms of Southern Africa, genus Natalina was split into 4 subgenera based on the work of Hugh Watson, who I'll come back to in a minute. We had Natalina sensu stricto here, and then Natalina afroritida here, Natalina capitina, and Natalina hyperritida. And one thing you need to note of these is they all have very long necks. And that's because inside the neck is a very elongate, very well-developed buccal mass, which is what controls the radula, the feeding organ, the ribbon of teeth inside the mouth, which the musculature is needed for the carnivorous diet. And here's one, I downloaded this from iNaturalist the day before yesterday. That is a long neck. And then the dwarf cannibal snails were divided into 2 subgenera, Nata sensu stricto and Nata natella, a very poorly known genus here, just one species in there, rather difficult to find, and as I say, very much less well known. So that was it in terms of the diversity. But what we wanted to do was to see what we could learn from studying the molecules. But before I get onto that, just to say why they're called cannibal snails, here you can see one is quite happy to take on one of the larger cannibal snails, African land snails. No problem eating those. May take a few days. They might carry the specimen, the prey item, back to their resting place and proceed to consume it over a number of days. They're also quite happy to eat the alien cornu aspersum. So from a perspective of gardeners, these carnivores are good to have in your garden because they get rid of the alien pest snails. And they're also quite happy to eat slugs. Even these brightly colored, highly mucousy, sticky things that one might assume are noxious, certainly to birds and so on. And it's really quite a quick process. It's a matter of 5 or 6 bites, maybe half a minute, and the prey's gone. It's eaten. And another strange thing that they do is they will often carry the empty shell of the prey item, carry it back to their resting place, and then they secrete acids from the sole of the foot, and the acids dissolve the shelly material of the prey item's shell, and that is then incorporated into their own shell, because they often live in habitats where calcium is in short supply. So they make use of every bit of the prey, including the shell. Just a comparison of the radula. I mentioned the radula. This is the ribbon of teeth that sits on top of the odontophore inside the mouth. It's like a tongue-like structure, and the radula is the organ that's used in feeding. In a herbivore, we have numerous very, very similar teeth. These are tiny, tiny teeth. It acts like a piece of sandpaper, gripping onto the vegetation and snipping it off against the jaw inside the mouth. The teeth are all pretty much of similar shape across the width of the radula, and very, very small, as I say. In these other carnivorous snails, the radula is completely different. Very many large, dagger-like or saber-like teeth, which are many, many fewer, but much, much larger. You can see these are almost well over 1 millimeter in length, each individual tooth. And these are used to grip and pull tissue off the prey items. Now, moving on to the hunter slugs. What's their distribution? Well, they are confined to southern Africa, ranging from the extreme western Cape here through the eastern regions up into southern Zimbabwe. That's it. They're not known from any other parts of the world. Very strange looking slugs. And they're not sticky and gooey like the Harionids and so on. they're quite dry and leathery to the touch and firm. They're not soft. Often they have ridges down the body, like these three here, often brightly colored. And you'll see the pneumostome, the breathing pole, is at the rear end of the animal. There's another one there. And they're somewhat reminiscent of testacella, but the difference here is that there is a shell, but it's internal. Testacella, as you will know, has a tiny little shell on the back of the slug, but with these, the shell is entirely internal, but it's still there. Why are they called hunter slugs? What do they eat? Well, many of them are subterranean, and they live in worm burrows and so on, and where they consume the earthworms. They are able to go into the burrows and follow the earthworms through the burrows and consume them underground. You rarely see them, but when the ground gets waterlogged, up they come, often, well, sometimes in the dozens. And that's obviously you need, if it's rained a lot, that's when you need to go out looking for these things. But it's not only worms that they eat. This is the ribbon, the radula ribbon of a hunter slug. Here you can see it's still wrapped around the odontophore, the feeding organ, and you can see again very large spear-like teeth that grip the prey. But as I say, they're not only do they eat earthworms, they will also eat pill millipedes. You have pill millipedes in this country, but in South Africa, pill millipedes are a good bit bigger. They can be the size of a golf ball. And they live under logs and fallen bark and so on in forest floor. And the slugs are also living in that kind of environment, and that's where the slugs are able to come into contact with the millipedes, and the slug will latch onto the back of a millipede, and the millipede appears to know that something is wrong. It realizes that staying rolled up in a ball is not going to help. Yeah, so they immediately start to open and close like this to try to dislodge the slug that is latched onto the back of the animal. But often to no avail. The slug is able to penetrate the thin cuticle between the plates of the millipede, the strong plates that are the millipede's primary defense, and they're able to inject some sort of toxin. We don't know anything about it, but it's able to immobilize the millipede, and you see here it's a lot of bubbles just beneath the head of the slug, and eventually the millipede becomes moribund. The slug lets go, and it moves round to the underside, crawls in between the head and the tail of the millipede, and proceeds to devour the contents of the millipede through the much thinner exoskeleton between the legs, and it consumes the internal contents of the millipede. So really a pretty remarkable feeding behavior for a slug. Now, I want at this stage to mention Hugh Watson. He studied both the chlamydophorids, the hunter slugs, and the Natalina, the cannibal snails, extensively over the years. And what I find remarkable about this fellow was that in 1915, he published this tome on the hunter slugs, and in it he drew comparison of the anatomy of the slugs with the cannibal snails, also drawing attention to the fact that there was considerable similarity between the anatomy of these South African species and those in Australia and New Zealand. And he invoked the theory of continental drift to explain this similarity, which, when you think about it, was way back in 1915, was only a couple of years after Alfred Wegener first got the idea of continental drift off the ground, and even then it was far from widely accepted. So Hugh Watson was insightful in terms of his interpretation of what he was seeing of the morphology. So what did we learn from studying the DNA? Obviously, we all know that there's a lot to learn from the DNA. And so we started by looking at the larger cannibal snails, and we found that we had our groups of what were previously recognized as subgenera, all coming out very, very nicely here as separate monophyletic clades, all with high levels of support here, 100% bootstrap support for those. But now we could see that really we're taking this in combination with the morphological information, that it really isn't appropriate to regard these as subgenera of Natalina, and it's actually much more appropriate to consider them as full genera. So that was the first thing that came out of that. We also found that this lineage here was not only genetically distinct from the others within Natalinus sensu stricto, but we also found morphological differences. So we created a new subgenus for that one within Natalina. Another interesting thing here is the other fourth subgenus, Hyperrhytida, which was originally part of Natalina. That we discovered was nothing more than just another species within the genus Afrorhytida. So this one, Hyperrhytida, the characters for that are just species level characters, and we now treat Hyperrhytida as a synonym of Afrorhytida. The distribution of these is as shown on this map here. Capitina is very much a Western Cape winter rainfall genus occurring in the Agalos region at the extreme southern tip of Africa. And Afrorhytida occurs in the winter, the all-year rainfall area here in the Western Cape and the Eastern Cape. And then Natalina, the largest ones of all, are widely distributed in the eastern regions, extending into southern Mozambique, but not extending further into Africa. And looking back at the morphology, we see that there are also consistent differences between these different genera in terms of the structure of the radula. Here we have the teeth are all very similar in shape, getting a little bit larger towards the edge and then smaller again. Here the teeth are very, very much larger at the edge of the radula, and in this one the teeth get larger towards the edge and then smaller. So the largest teeth are some distance in from the edge of the radula. And there are also differences in the structures of the spermatophores. Little tadpole-shaped ones here, spiky ones here, and smooth, velvety ones in Afrorhytida. So for our molecular work, we did these was the larger cannibal snails. The obvious thing now was to add in some others. So we added in the dwarf cannibal snails. We added in some Australasian rhytidids, and we wanted to put in the hunter slugs to see where they fitted into the picture. This was the resultant tree that we got. And we see we have our Natalina sensulato clade here with Natalina itself, Afrorhytida and Capitina. And then we have Nata here as a single entity, well supported here, a monophyletic radiation. But Natella, the little odd one I mentioned, is now on its own up here. So that was an interesting discovery. Basically, Natella is not part of the Nata lineage, and so we are now recognizing Natella as a full genus in its own right. And then we have the Australasian ones here, as a cluster together as well. But the chlamydophorids, supposedly a different family, are sitting within this thing. So the similarities that Hugh Watson saw in terms of morphology are also reflected here in terms of the genetics. And the relationship between Chlamydophorus and this Natalina sensulata clade is very strongly supported here. So this is a reliable relationship. We can consider it a pretty conclusive finding. Here we are in more detail. Chlamydophorus as a clade in its own right, well supported. And then the Natalinas sensulata, also a clade on their own. And the whole, the sister group relationship here between these two is also well supported. Same with Nata. Considerable divergence in terms of the genetics here. But an important thing to remember or to see here is how similar all of these are. There's very little morphological differentiation. We've got morphological stasis, but we've got considerable genetic divergence. If we go back to look at this lot, you can see how different these all look and how similar all those look. And then again, with the Australasian ones, there's a good deal of morphological diversity reflected there. But we can't look at this in too much detail because there are so many taxa within this Australasian right-titted group that we have just a small fraction of them here. And it's difficult to make any conclusive remarks when you've got so little representation. So here we have the one cluster, Natalina sensulata and Chlamydophorus. Here we have the Australian ones, and here we have Nata, with strong support for key nodes here. But another point to look at here is that we actually have, for these deepest nodes, we have low levels of support. So we can't, this topology that we're looking at here is not reliable. The strength of support indicates that the positions of those things could change. And this is a perennial problem at the moment with the phylogenetics of land snails, is that the deeper nodes are difficult to resolve, difficult to get reliable support levels for the topology. So, as I've said, we have Rhytidity and Chlamydophoridae in the same clade. Two family group names in one clade. We can't have that. So we have to decide which name do we use? Well, Rhytidity, the name, stems from Pillsbury in 1893, and Chlamydophoridae stems from Cockerell this is a difficult one. Chlamydophoridae was proposed by Cockerell in 1935, but as a replacement for a name that was based on a synonym, a junior synonym. So it actually, from a priority, from the priority perspective, it takes its name here from 1903, the date of the earlier name, based on the synonym. So either way, that is not relevant because both of these dates are later than Pillsbury's name. So the family we must use for this family name is the Rhytidity. So we accept Rhytidity, but within this group as a whole, we can see we have two sections, two large groups. The Chlamydophorus and Natalina, and then Nata and the Australasian ones. And the genus Rhytida is in here, where Rhytida here, on which the family name is based. So if we're going to think of these as subfamilies, we take Rhytida there for the Rhytidini, and this now becomes, with Chlamydophorus in there, this becomes the Chlamydophorini. So we have the family Rhytididae with two subfamilies, Rhytidini and Chlamydophorini. And now, looking back on the distribution as a whole, I said we had this problem with Madagascar. In 1990, Ken Emberton, an American malacologist, proposed that there was a lineage in Madagascar that he thought, or a species that he thought was actually a Rhytidid. And it's a very attractive proposition to want to find a Rhytidid here in Madagascar, another fragment of Gondwana. So we decided that we would like to examine this further and test out this hypothesis that there is a Rhytidid there. So we had to find it. The species concerned was described in 1879, by E.A. Smith as Helix coveni. And for a long time, it had been referred to the family Acarvidae and placed in the genus Ampelita. But now Ken Emberton proposed that it should be placed in Rhytida. And if you compare it with a South African Rhytidid here, Afrorhytida krausi, you can see that there's remarkable similarity in the shells. They look very, very similar. So his suggestion or hypothesis was not without foundation. Where does it occur? Well, it occurs on the high altitude mountains of the eastern regions of Madagascar. There's a ridge of mountains that runs up this whole central eastern region, and the climate there is very, very damp, rainforest type environment. And we've been doing some survey work over here in the northwest of Madagascar, and on our way back we decided that we would try to visit the forests here in the Andasi Bay region to see if we could find Helix coveni. Glorious territory, wonderful high altitude rainforest. It's the Maramizaha forest is near Andasi Bay, and if you've ever been to Madagascar, you may have been to see the lemurs called Indris, and this is the kind of habitat and the region in which they are to be found. But it's not fun collecting there. It's wet. It was jolly cold, and we struggled a bit, and we had to wade through huge areas of habitat transformation where we could hear the chop, chop, chop of the locals cutting down the forest to burn the wood to make charcoal, which they sell on the side of the road, and it's taken away to Antananarivo and Toamasina and so on for people to use to as a fuel source for cooking their food. So it was that was really quite an alarming and disturbing sight. Anyway, we found it, just the one, but one is enough when you're talking of DNA. And so we were pleased with our findings. And the question was, is it a Rhytidid? Well, we could look at the structure of the embryonic shell and the adult shell. We could look at the external anatomy, the pulmonary anatomy of the distal reproductive tract, and the structure of the radula and the jaw, and we could also look at the DNA. So the radula and the jaw first. This is it for Helix coveni, and immediately you'll see that we have a very typical herbivore radula. Lots and lots of very similar teeth, little differentiation between the teeth, except perhaps at the edge of the radula you get some differentiation there. And the jaw is what we call an olecognathous jaw, little scratch-like grooves across the width of the jaw. Comparing it with the radula jaw of a known acarvid, Ampelita namarocoensis, and we see very much the same. Lots of tiny, tiny teeth, a little bit of differentiation towards the edge, and another olecognathous jaw. If we were looking at a right-titted, this is the sort of structure we would anticipate, as I've shown you already, very many large teeth, dagger-like teeth, saber-like teeth, completely different radula structure. And of course, with a right-titted, there is no jaw. So looking at the pulmonary anatomy, this is one of the lovely drawings that Linda Davis has done. Here we have the heart here with the auricle and ventricle. This is the kidney. And then from the kidney, the ureter runs out first, the primary ureter to the posterior of the pulmonary cavity, running forward as a secondary ureter here. And we see that only the very beginning of the primary ureter is closed over with a little hood here. The bulk of the primary ureter is open. If it was a right-titted, the whole of the primary ureter would be covered over, and only the secondary ureter would be an open channel. So the morphological evidence is telling us that we have an acarvid here, and that it might actually be genuinely a species of Ampelita. The DNA. So what we did here was we had the information from the molecular phylogeny for the Stylomatophora from Christopher Wade here in the UK, and we took the information in there and we added to that genetic data for additional right-titted genera from Australia and New Zealand and South Africa, as well as some representatives of the Madagascan acarvids, all the recognized genera, and we added also some caryodid species from Australia, and into that mix we popped Helix coveni as well. And what did we find? Well, we found that here's our right-tittedy in red here, and here's our acarvidae in green here, and here is Helix coveni. There. So again, pretty conclusive evidence from the DNA that sadly we don't have a right-titted in Madagascar. Helix coveni sits fairly and squarely within the acarvidae. We can't say exactly where. All we know is it sits within the Madagascan radiation of the acarvidae. Acarvus phoenix is from Sri Lanka. But Ampelita itself, there's more than 100 species of Ampelita. So in order to refine this further, you're going to have to put in more different species. But nonetheless, we got the information we needed. So we now say goodbye to Madagascar as part of the right-tittedy distribution. Now, I hear you say, you're talking Gondwana here. What's happening here? This was also part of Gondwana. Why have we not got any right-titteds over here? And we can get around that one by not looking at the right-tittedy, but by going up a level, and we're looking at the Rhytidoidea, the superfamily, rather than the family. And if we do that, then we see we've got 3 or 4 families of Rhytidoidean snails in South America. We've also got the acarvids. They're also part of this superfamily in Sri Lanka, Madagascar, and the Seychelles. We've got the caryodids in eastern Australia, and we've got the dorcasids in the drier parts of western South Africa. So at this level, we now have the coverage we wanted for Gondwana. So it's just a matter of where in the hierarchy you ask your question. Now, moving on to Prestonella, this funny little group of snails that are now known as the Karoo rock snails. They had no common name until recently. This is the type species, initially described as Bulimus bowkeri, now Prestonella bowkeri. So a pretty nondescript looking snail. Here are some photographs of living examples. There are 3 described species. 2 of them we found alive. The third one is still a bit of a mystery, but they don't differ from each other particularly much, and they occur in similar kinds of habitat. This is the known distribution. We have Prestonella bowkeri here from Somerset East, and then another lot over here to the west of Beaufort West in the Karoo. And then we have Nuptialis, Prestonella nuptialis here in Craddock, and also there's a population here in Somerset East. And the third species, the poorly known one here in southwestern Lesotho. We've looked for it there, but we have not found any evidence of a Prestonella-like thing in that part of the world. So this was our distribution pre-2000. Prestonella was originally placed in the family Bulimulidae by Connolly when he described the genus. And in fact, the Sowerby, when he described the species, put it in Mesembrinus, or thought it might be a Mesembrinus, which is also now recognized as a Bulimulid species, or genus, sorry. But subsequent to that, people working on this family stated that Bulimulids are not thought to occur in Africa. So Prestonella was removed from this family. Very little material of these snails have been collected, and none during the 50 years before we started to think about this question. I've shown you the shell. The shell features are not really helpful. It's a nondescript little brown job. It's been suggested that it belongs to the family Aeidae, also Succineidae or Eurocyclidae, and perhaps some people have even suggested it's a family on its own. So it's been a long-standing enigma for many, many years, and you can view such a thing as a headache or as a challenge. Obviously, if you consider it a headache, you're not going to make any progress. But if you view it as a challenge, then clearly you've got to investigate it. To do so, we needed live material, ideally of the type species, from the type locality. Type species, as I said, Prestonella bowkeri, the type locality of which is Glenavon in Somerset East. Glenavon is a farm in Somerset East, and I utilised contacts in the Eastern Cape. Mary Cole, who works at the East London Museum, and she knows the Eastern Cape like the back of her hand, and she was able to establish exactly where Glenavon was. Sadly, there was no precise habitat data available with the specimens, but some of them did say from the gorge in Glenavon, gorge at Glenavon, Somerset East. Now, there is a gorge at Glenavon, Somerset East, and there's a nice waterfall that falls into the gorge. So we launched Operation Prestonella in 2002, and our goal was to find this beast alive. So I went down there with Mary Cole and headed for Glenavon. This is the gorge here. You can see a high level, what's called the Bosberg, the Bush Mountain, and there's a river course, a water course on top, which plunges over the escarpment edge into the gorge down towards Glenavon Farm, which is over here somewhere. So within an hour or so of arriving there, we found it. We hit the bullseye. And there they are on the rock surfaces, not in the water by the waterfall, but close to the base of the waterfall. So there's high levels of humidity here. We're talking of a waterfall that's 100 meters high, so it's a good fall. And we also found it here at the top of the watercourse. There's also a bit of a gorge up here with rocky walls on either side, and we found them there too. So we were jolly happy with ourselves on that score. The next day, we went to see if we could find the other species, Prestonella nuptialis. And it was described from Elandsberg. And we also had a little bit of habitat, microhabitat data here, because it said on some of the specimens in the KwaZulu-Natal machine, found in crevices between the rocks on top of the Elandsberg. So we got to the Elandsberg, and I thought, don't tell me we've got to get up there. So I thought, oh, there's more than one Elandsberg near Craddock. So we headed for the other one. I haven't looked at this one. We headed for the other one. The other one was even worse. So we came back to this one. And we managed to locate the and we asked him if we could go on his land and get to the top. He was only too delighted. So we launched off to get up here. It actually wasn't as bad as it looks. And of course, it's scenically stunning. I mean, who doesn't want to take a walk there? So we got to the top, and as it said, on the top of Elandsberg. Well, we hunted around on the top, and we didn't find any evidence of it all. But then on the way back, on the way down, just below the summit, there's these rocky crags and crevices of cliffs, as it were. And we started looking in there, and sure enough, we found them there. They like rocky surfaces, these things. In subsequent days, we went to other inselbergs like this around Craddock, and we found more of them. So this is clearly their habitat. So we had our specimens from the type locality, both species from their type localities. And again, we went back to Christopher Wade's molecular work. Now, this time, this was before the 2006 publication, so we were using his 2001 data. Doesn't really matter. It covers a broad spectrum of families from around the world. It's based on mitochondrial and nuclear markers. And our aim was just to rerun the analysis. Chris Wade was generous enough to let us have his data. It had already been published. So we just wanted to pop the information from Prestonella, the genetic information, in with his data and rerun the analysis to see what told us. And then after that, we were going to look more at the morphological information to see if we could get confirmation of whatever the molecular data told us. So this is Christopher Wade's phylogeny. Stylomatophora here marked out in red, 100% support for that. So popping Prestonella into this equation, it came out here. Here's looking at that in more detail. Prestonella arrowed there, next to Bothriembryon indutus and two species of Placostylus. Placostylus are those large snails from northern New Zealand and Melanesia and so on. And Bothriembryon is an Australian genus. And next to that, also another clade is of Bulimulus, which is an old South American species. So here we had African Prestonella nestling between Australasian genera and South American genera. I confess I was pretty flabbergasted that our little snails from the middle of the Karoo were most closely related to these things from Australasia and Melanesia and so on. So we went on to look at the reproductive anatomy, and sure enough, there was a good deal of similarity here. In terms of the distal genitalia, there are no accessory structures associated with it, no dart sacs, no mucus glands, no seci or stimulators of any kind. It's a fairly simple pulmonate reproductive system. There's limited differentiation between the penis, epiphyllus, and the flagellum, and those three structures are very long. And the penial retractor muscles attached, this is the body wall here, The penial retractor muscle is very short and attached to the very tip of the flagellum, all characteristic of members of the Bullamulidae. The vas deferens is also very thin-walled and partly embedded in the wall of the penis. Looking at other organ systems, the embryonic shell, again, this tilted angular protoconch here with strong axial sculpture is entirely consistent with a Bullamulid relationship, as is the radula. Radula doesn't tell you a lot. The herbivorous groups, the radula is usually very similar. But the jaw too, not Olacognathus like the last ones. This is a Stegognathus jaw. It's made of overlapping plates rather than little transverse grooves. And in terms of the pallial organs here, in the pallial cavity, again, the heart, and here, the kidney. And here we see the primary ureter and the secondary ureter are closed for almost their entire length, only opening just inside the pneumostome here. So again, all these things fully consistent with the position in the Bullamulidae. So the distribution of Bullamulids pre-2000, the statement, no Bullamulids are present in Africa, Brambra, 1979. Well, I think he's wrong. He won't be upset to hear me say that. He fully agrees with our findings, and he's very happy, as we all are, to find this. Now we've got a Gondwana, classic Gondwana type distribution for this family with the New World, and then we've got Southern Africa and Australasia here as well. So we started off with Connolly placing Prestonella in the Bullamulidae. Sowerby too had placed Prestonella in a Bullamulid genus. And now we've come half a circle. We're in limbo for 50 years. And then now we're back again in the Bullamulid. So we have one circle completed there. But now another little circle. I digress here, but another circle. Matthew Connolly, who named the genus Prestonella, he was in South Africa for a very long time, but his collection is now here at the NHM. Here is Hugh Watson, the fellow I told you about before, and we ask, why did Connolly place Prestonella in the Bullamulidae? The answer is that there was some live material, or live taken material, preserved material of Prestonella available. And Hugh Watson had studied it, and he had told Matthew Connolly about his findings and had said, this thing that you're working on is a member of the Bullamulidae. And Connolly stated that as much in the paper in which he published this genus. But he said Watson was just about to publish his results. Well, Watson never did. So we have never been privy to Watson's findings. Now, another little addition to this is this fellow, Henry Burnap. Henry Burnap was born and brought up in Newcastle. He studied in Edinburgh, and then he emigrated to South Africa in the 1870s. And he ended up in Pietermaritzburg, and he developed a passion for mollusks. And particularly he collected marine snails and also terrestrial ones, but terrestrial ones were his particular passion. He developed an extensive collection, and he communicated with leading people all around the world, Pillsbury in America, Belleville and Ponsonby here in the UK, and he became the sort of go-to person for information on South African mollusks. And as I say, he lived in Pietermaritzburg, and he became, when they formed the Natal Museum around about the turn of the last century, around about 1900, the museum was formed, and Henry Burnap was brought in as the honorary curator of mollusks at the fledgling Natal Museum. And he stayed there for a very long time, practicing as an accountant professionally. Eventually he retired, and he gave even more of his time to the museum and the malacology collection. And then he eventually gave his collection to the museum, and he worked there as honorary curator till his death in 1928. And that collection formed the nucleus of the collection at the KwaZulu-Natal Museum. Now, the interesting thing here is that Henry Burnap was Hugh Watson's uncle. I don't know if you can see a bit of similarity, or whether it's just a goatee beard or whatever, but maybe there is some similarity there. They're also evidently quite similar people. They were difficult to get to know, very fastidious. I think people were terrified of Hugh Watson because he was such a stickler, but his anatomical morphological studies are exemplary. And Henry Burnap, I think, had a similar sort of mindset. So Henry was Hugh's uncle. And as I say, it was his collection that formed the basis of what is now the KwaZulu-Natal Museum collection. And this is the largest collection of snails or molluscs in Africa, and one of the largest in the Southern Hemisphere. And it was here that I worked for 33 years with Tamsin's dad. And now here I am today talking to you about Prestonella. So the distribution of Prestonella pre-2000, the distribution now, we can see that they're all along the southern edge of the Great Escarpment in South Africa. And many, many more localities we found them. And they're always associated with river gorges, rocky walls, and such like. Also associated with the edges of escarpments like this here, and this Inselberg type habitat. It's great fun to work in places where the scenery looks like that. But now you might look at that and think, it's harsh up there, and it is. But if you go there on a day that's not quite so sunny, you often find that the edges of the escarpments and the summits of the Inselbergs are covered in cloud. And it is this that enables the snails to survive there. There's obviously, whether it's raining or just misty, there's obviously moisture available, and the snails are able to survive in what is otherwise a pretty dry environment. It might remain perfectly dry in the surrounding matrix, but the summits of these Inselbergs and the edges of the escarpments are receiving some precipitation of some kind. Next question, are they threatened? My initial or instinctive response was, yes, these things must be threatened. They live in such highly specialized snails, living in such small, restricted habitat. They must be threatened. But I remember talking like this to a group of biologists in South Africa. One of the botanists said to me, why do you think they're threatened? These are survivors. They've been here since the time of Gondwana, They're tough little things. It's not sort of, doesn't follow on necessarily that they must be threatened. And I thought to myself, he's got a point. Yeah. So we did some additional experiments to try and establish or to look into their threat status a bit more closely. So a student working at Rhodes University in Grahamstown in South Africa, he did some mark and recapture studies here at the Glenavon population, looking at them here on the rock walls beside the waterfall. Here they are. You can see some numbers, but at first sight, he went there, he counted them, and his original estimate of the population size was less than 100 individuals there. Then he did his mark and recapture experiments. He painted them blue. He went back repeatedly and counted how many of the marked snails he found. And by the time he'd finished, he estimated there were perhaps more than 4,000 individuals. at this one particular site here. So there are a lot more of them than meets the eye when you go there. They're hiding away in crevices and inaccessible places. So they're not as scarce as one might imagine. So maybe they're not as threatened as we first thought. But I still believe deep down that if climate change progresses as it is, things are not looking good for these snails. If these watercourses dry up, if the climate dries out to such an extent that these inselbergs and the escarpment edges no longer get clouds forming on them, then what is the future of these snails? I think it is a matter of concern. Just finishing off, in the meantime, this was all done 20 years ago. Our studies on the where Preston leather fits into the picture, and we came out with the answer Bullamulidae. Well, it's no longer in the Bullamulidae. This group has been studied much more extensively now, and the superfamily that the Bullamulidae belongs to, the Orthalicoidea here, has been divided up much more finely, and the Bullamulidae has been restricted to a purely South American lineage here. And now we recognize the family Bothriembryontidae, and within the Bothriembryontidae we have the Bothriembryontinae from Australia and Melanesia, New Zealand, and so on. So that's Placostylus and Bothriembryon, and then a rather ill-defined cluster here, sort of a paraphyletic entity known as the Prestonellinae, sits here, composed of South American genera and Prestonella itself. But the levels of support for some of these lineages is very low, so the actual topology of this thing might change. But the interesting thing about this is that this lineage is the only family within the Orthalicoidea that is actually present across almost all the Gondwanan fragments. The other lineages are mostly in South America. And if you look at them, living representatives of these genera, you see that they're actually quite, they look quite similar, superficially at least. So that they all belong to one family is perhaps not as surprising as you first think. So we're almost at the end of my presentation now, and I want to show you a picture of something that if you are a snail or an earthworm, you do not want to see, because it means your end is nigh. This is the head of Natalina cafra coming up, and you are about to meet your doom. They have these wonderful palps here, which they use to sense the ground. They're moving him all the time, testing out for the trails of earthworms and the trails of snails. And yeah, it's not looking good if that's coming up upon you. So it just remains for me to thank, I mentioned Linda Davis there. She accompanied me on many, many field trips, and she did an awful lot of the drawings. And Mary Cole, too, of the East London Museum, has played a considerable role in helping us with these studies, and the names of some of the other people. So I'll leave it there. Thank you very much. Any questions? I'm happy to answer.
Brilliant. Thank you, Fran. I'll stop sharing that so we can see if anyone's got any questions from at home as well. See one from David Holly up there. Will you start with him? David?
I didn't actually flag up that I was going to ask a question, but one that immediately springs to mind now is how well, Dai, do you think the South African land snail fauna is currently known at species level? I guess a faunal inventory is fairly complete, but it would be interesting to have your view on that, particularly perhaps with respect to micro snails and other more tricky customers. Compared to tropical Africa, clearly most of tropical Africa, South Africa, it must be very well known, but it would be interesting to get your perspective on it.
The South African land snail fauna is, compared to that of tropical regions to the north, is pretty well known. But there are still some really problematic areas. The subulinids are one area, the endodontids too. There are many species that, or there are many things that probably represent new species, and we need to investigate them more fully. It's evident that there are many genera within the cheropids that are remain to be described. So there's still an awful lot of work that remains to be done on the South African land snail fauna. We're continually finding new species of Gulella in restricted range habitats. So it's well covered relative to tropical regions, but there's still loads to do.
Yeah.
And there are areas too, geographically, that are poorly known. Parts of the Cape remain really insufficiently sampled. The eastern regions are perhaps where I was and where most of the work has been done, where Henry Burnap did his collecting. Those are perhaps better studied, better collected, but there are big areas within the Karoo and in the western regions and even in the Cape Floral Kingdom that require an awful lot more survey work. And also I'd say that the conservation status, the red listing status of many of the snails needs to be looked at in more detail.
Thank you.
Anybody else?
Heike and Hutch.
Yeah, okay. I have a question. On one of the pictures on your hunter slugs, one could see the pneumostome quite well.
Yeah.
And it looked a bit as if it was slightly on the left side. Is it?
Not really. No, it's pretty much in the middle.
Yeah.
There's a bit of, some of them have almost a bit of a plate or flattened area on the back, and it's usually smack in the middle of that.
Yeah.
With
So
the,
it just
the
looks like
skin
that.
grooves leading into the pneumostome there.
All right. Thank you.
I have a question about those slugs as well. So you describe them as being dry to the touch.
Yeah.
And are they very slow moving as well?
Oh, slow, like you can't believe.
Like Testacella is like that.
Yeah.
And I can't understand what it gains from being dry and slow. How does that help it catch worms or live without a shell? What do you think?
Well,
You've obviously evolved these things completely coincidentally or convergent.
yeah. I think they the ones that live buried underground are obviously not subject to desiccation to the extent that the ones living on the surface are. And the ones that live on the surface are generally in forests. They're in damp environments. They're very, very cryptic. They're not like some of the other slugs which sit out on leaves and so on. And they benefit from being sticky, like you can't believe, and probably tasting foul. So, as I say, the slugs are very cryptic. Slow moving. I think they must be amongst the most slow moving animals on the forest floor. They really do move very slowly. But how they catch living prey, then, that doesn't make sense either. They find them under logs, I think, is the thing. The prey is stationary, resting up, and then the slugs are able to find them. Any more?
Yeah, good question from India.
Yes. I lived in South Africa for 3 years in the 60s.
Uh-huh.
And we had a Zulu cock. And as a little person, he'd been out in the bush, I think probably near Pietermaritzburg, looking after the cattle. And they'd had to live off the land, so they ate the snakes. Would they have eaten the snails?
I'm not aware that that's part of the local culture of Africans in South Africa.
See?
Yeah, they eat snails are eaten extensively in West Africa, the acatinids, yeah, very much so. But I'm not aware of them being eaten in Southern Africa. Peter, do you know if they or Ben, are they eaten in East Africa?
East Africa, the Ugandans eat.
You
The
know,
large
Uganda.
and large.
The acatinids, yeah.
The Acatinids.
Yeah. No, they don't seem to be used so much in as a dietary item in South Africa for sure.
Some poisonous.
I don't think acatinids would be poisonous. I wouldn't like to eat these cannibal snails. No, I don't know that they would be good to eat because I think that there are substances. They have a well-developed pedal mucus gland, and I think that is involved in some of their feeding behavior. There are noxious substances produced by that. I think also the hunter slugs also have a similar well-developed pedal mucus gland, and it might be this that is producing the toxin that is gets into the millipedes that they're eating. Because there certainly seems to.
I mean, they have predators.
Oh yes. No, there are predators. Mongoose will eat them.
Mongoose.
Yeah, for sure. Yeah,
Yes.
and things like bush pigs and so on. The bush pigs will eat anything, so they will munch acatinid snails. They'll munch the anatolinas as well. So there are quite a lot of predators that will eat them.
So no wonder they hide in cracks.
Well, yeah.
I bought a couple of snails from Sri Lanka. From the genus Corilla.
Gorilla, yeah.
And in MolluscaBase, it says they're vitidins. But I don't know.
I'm not sure whether coryllids are they still. There's some uncertainty as to where they belong now, if I remember correctly. Yeah, I know the Sculptarias are no longer considered part of the coryllids, and they are removed from the Rhytidoidea these days. Yeah, but the coryllids I'm not absolutely sure about. Yeah, but certainly those South African Sculptarias used to be considered coryllids, but no longer. Yeah.
Yeah, these things are changing all the time.
They are, yeah, and then there are these difficult groups that are only associated with superfamilies through very tenuous links through these deep nodes in the tree. Yeah.
There was a message in the chat there about ancient humans, I think. Ancient humans ate giant land snails, according to new research. Border Cave, South Africa.
Oh, well, there you go.
I don't know how ancient that is, though, because a lot of the earliest hominid stuff is from Africa, goes way back.
Oh, certainly,
Homo.
in the archaeological context, there are often the remains of snails found in the deposits in caves and things like that, and one can only assume that these items did form part of the diet of these people from many years ago. But the inhabitants of the local inhabitants, indigenous people of eastern South Africa today, I think lead a very different kind of existence. And I'm not aware that snails are a regular food item for them. They may use them for traditional medicine purposes, but rarely, you don't see them for sale in the markets and things like that, for example.
Okay. 200,000 years ago. So that was a long way back.
Yes. Yeah.
That's before early modern. Don't quote.
Yeah.
Right. That's absolutely fantastic, Dai. Thanks for making not just the snails, but also the whole sort of landscape and the history of it come alive with some brilliant examples. And will you come back and do the marine side for us?
I don't know that I've got such interesting stories on the marine side. Yeah.
And the pictures are pretty as well.
Oh yeah, the marine
snails
one. Yeah.
even prettier than the ones at the moment. Yeah. Okay, right. Thank you very much.
That's it.